Students, in this session we are going to discuss a few bacterial plant diseases. We will be discussing bacterial leaf blight of rice, citrus canker, crown gall and angular leaf spot of cotton. The first one is bacterial leaf blight of rice. The causative organism for this disease is Xanthomonas oryza. This is an aerobic, gram-negative, non-spore forming rod which occur either singly or in pairs. In nutrient agar, this organism will form pale yellow, circular, smooth colonies with an entire margin. This organism causes bacterial leaf blight of rice, in which there will be wilting of seedlings and yellowing and drying of leaves. This disease will cause a yield loss of up to 70%. Bacterial leaf blight of rice is a vascular disease. That means the bacteria enters the plant and moves through xylem. The primary sources of this infection is diseased seeds and stubbles which are present in the field. And secondary infection spread will occur during wind-borne rain splashes, and during this the pathogen, the bacteria, will enter the host plant through wounds and stomatal openings. The bacterial leaf blight of rice may either occur as seedling wilt, which is also known as cresek, or leaf blight. So what is seedling wilt or cresek? It is observed in the rice plant, in the rice seedlings 1 to 3 weeks after transplanting. There will be formation of green water-soaked layer along, which can be observed along the cut portion or in the leaf tip of leaves, and this is the early symptom. Then the leaves will wilt and roll up and will become grayish green to yellow colored. During seedling wilt, the entire plant undergo wilting. Coming to leaf blight, here the symptoms are there will be formation of water-soaked greenish to yellowish stripes on leaf blades, or which may start at leaf tips. There will be undulated yellowish white or golden yellow marginal necrosis in the leaves. This will lead to drying and curling of leaves. There will be a bacterial ooze out from the leaves which looks like a milky or opaque dew drop on young lesions, which is generally observed early in the morning. Severely infected leaves will dry up, and this disease results in a loss in grain yield, generally up to 60 to 70%. So the disease in seedlings is known as seedling wilt or cresek. In more mature or bigger plants, the disease is known as leaf blight. So this is a paddy field affected by this particular disease, and this image shows a leaf affected by leaf blight. Coming to the epidemiology and factors favoring the disease development, the following are the factors which favor disease development: presence of weeds, presence of rice stubbles and ratoons of infected plants in the field, the presence of this particular causative bacteria in the rice paddy and irrigation canals. A warm temperature and high humidity favors the disease development, and if there is continuous rain and deep water, it also predisposes towards disease development. Severe winds, over-fertilization, etc. are also factors which favor the disease development. The use of trimming tools and handling during transplantation can also trigger new infection in the field. For the management of disease control, for controlling the bacterial leaf blight of rice, there are various strategies adopted, and they include seed treatments. The seeds may be treated with bleaching powder and zinc sulfate, or it may be soaked for 8 hours in agromycin and vegetable ceresan, followed by a hot water treatment. The seeds may be soaked for 8 hours in ceresan and with streptocycline. Spraying neem oil or neem seed kernel extract in the field also helps for disease control. Another method is the spraying of fresh cow dung extract, which may be prepared by suspending cow dung in water and after allowing to settle for some time, the supernatant liquid may be sieved out and sprayed in the field. And this treatment should start from the initial appearance of disease and then it should be repeated at fortnightly intervals. Cultural methods can also be changed for the management of the disease. It includes the growing tolerant or resistant varieties of rice. By growing the seedlings, preferably in isolated upland conditions, a balanced fertilization by avoiding excess nitrogen is essential for proper disease control. Draining the field, destruction of weeds and collateral hosts, avoiding flow of water from affected fields, maintaining proper plant spacing and chlorination of irrigation water. All this will help in proper disease control. Burning of infected straw and stubble should be done to destroy the bacteria which are present in the field, in the affected field. Spraying of streptomycin sulfate tetracycline combination with copper is to be done in the field. Bleaching powder application in the irrigation water helps to inhibit or control the disease in the crusade stage. Foliar spray with copper fungicides alternatively with streptocycline also helps to prevent disease spread further. So this is about bacterial leaf blight of rice caused by Xanthomonas oryzae. Now we are going to discuss the second bacterial disease of plants, citrus canker. The causative agent for this disease is Xanthomonas axinopodis. Xanthomonas axinopodis is an aerobic, rod-shaped, gram-negative, capsulated bacteria having monotrichous flagella. The colonies of this bacterium is yellow colored due to the presence of the pigment xanthomonadin. When this organism is grown in a culture media containing sugars, then the colonies will be very mucoid due to the production of an exopolysaccharide slime. For the identification and classification of bacterial isolates into different pathovores, there are different types of pathovores of Xanthomonas axinopodis. For this purpose, serology, bacteriophage typing, fatty acid profiles, PCR, DNA analysis, etc. are done. And for bioassay, detached leaves or leaf disc is used. Xanthomonas axinopodis cause citrus canker in numerous species of citrus and citrus relatives, including orange, grapefruit, pomelo, mandarin, lemon, lime, tangerine, etc. This disease significantly affects the vitality of citrus trees, causing the leaves and fruits to drop prematurely. The fruit infected with canker is safe to eat, but it is generally unsightly. Coming to the symptoms of citrus canker, there will be formation of characteristic lesions on leaves, stems and fruits, and these lesions are generally raised, brown with water-soaked margins, usually with a yellow halo or ring effect around the lesion. This disease is mostly a leaf spotting and fruit rind blemishing disease, and it also causes defoliation, shoot dieback and fruit drop. So, We will discuss about leaf lesions first. The leaf lesions start as pinpoint spots and attain a maximum size of 2 to 10 millimeter diameter. The leaf lesions will become visible about 7 to 10 days after infection, and these are observed generally on underside of the leaves, and thereafter they will begin to form on upper surface as well. The young lesions are raised or pustular on both surfaces of leaf, and these pustules then become corky and cratery-form with a raised margin and sunken center. There will be a yellow halo that surrounds these lesions and water-soaked margins around the necrotic tissue. The fruit and stem lesions are also formed. The fruit lesions generally extend to about 1 millimeter depth, and these cause premature fruit drop. Generally, the internal quality of fruit is not affected, but occasionally the lesions may penetrate the rind and expose the interior of the fruit, and this will result in secondary infections by other microorganisms, which may lead to the decay of the fruit. The stem lesions, they will remain viable for several seasons, and these are the lesions in which the bacteria can remain for a long term and support the long term survival of the pathogen. So, these are the leaf lesions and fruit lesions. So, about the disease cycle and epidemiology, the bacteria survive and multiply in lesions in the leaves, stems and fruits until they upsize and fall to ground. The bacteria is capable of surviving in lesions on woody branches up to a few years of age. The bacterial ooze that comes out from the lesions onto plant surfaces, the bacteria will die rapidly upon exposure to direct sunlight and also due to drying. If there is free moisture on the lesions, then the bacterial ooze out will remain there and the bacteria will get dispersed to start new infections. Wind driven rain along with wind is the main dispersal agent which aids in the penetration of bacteria through stomatal pores or wounds present in the plants. The wounds may be either made by thorns, insects or by blowing sand. Pruning also causes severe wounding, and the bacteria can enter the plant through these wounds and leads to infection. The infection occurs on leaves and stems within the first 6 weeks of growth initiation, and the mature leaves, stems and fruits are generally resistant to bacterial infection, and they will get infected only if there are wounds in these leaves, stems or fruits. Spread of this organism, the bacteria, for short distances are generally carried out by wind and rain. And since it is carried out by wind and rain, the disease develops more severely on the side of the tree which is exposed to wind-driven rain. The disease spread over longer distances occur due to severe meteorological events such as tropical storms, hurricanes, tornadoes, etc. So how to control and manage citrus canker? There are 3 approaches: exclusion, sanitation and eradication. Coming to exclusion, there is this particular disease, citrus canker, in some countries. This is because of the importing restrictions on propagating material and fruit in these countries from other areas having the citrus canker prevalent there. Sanitation means the new infections of citrus canker are generally related to human and mechanical activities. So, by constructing decontamination stations for personnel, vehicles and machinery, the infections from human and mechanical activities can be prevented. Proper sanitation inhibits the disease progression. Eradication means the removal and destruction of infected and exposed trees. Thereby, the bacterial inoculum, the bacterial pathogen, may be eliminated from an area. The trees have to be uprooted and burned down or cut down and chipped so that the bacterial transfer from these infected trees is prevented. So, for the control and disease management, complete destruction of the diseased plants has to be done by burning them. Disease-free nursery stock should be used for planting. Also, resistant varieties, if available, it would be better to use the resistant varieties of citrus. Spraying the plant before planting with 1% Bodex mixture will help to inhibit the growth, thereby inhibit the growth of the bacteria, thereby inhibiting the disease occurrence. Antibiotic spray and spraying the leaves with neem cake also helps to inhibit the disease spread. And it is better to avoid working in infected orchards when trees are wet from dew or rain. And the disease spread may be minimized by the reduction of wind, by setting up windbreaks on the perimeter of the orchard or between rows. The next bacterial disease is crown gall, which is caused by Agrobacterium tumefaciens. This is a gram-negative aerobic flagellator rod which survive in soils with good aeration. These bacteria are also present on the surface of roots. Thousands of plant species are susceptible to the infection by Agrobacterium tumefaciens, and these plants include plants belonging to the family Rosaceae, shade and nut trees, many shrubs and vines including grapevines and perennial garden plants. All these are susceptible for crown gall disease. And during crown gall disease, there will be the formation of roundish, rough surfaced galls or woody tumor-like growths which are having several centimeters or more in diameter, and these are generally formed in the plant at or near the soil line or on roots and lower stems. It may also develop on a graft site or a bud union site. The galls are first cream colored or greenish and later the color turns to brown or black. The plants having crown gall lose vigor and eventually die. The symptoms of crown gall first appears as small overgrowth on the stem or crown or roots near the soil line. Initially, the gall or tumor is white or flesh colored and it is more or less round and have a soft or spongy texture. The gall slowly enlarges and develops an irregular, convoluted, rough, corky surface and a hard woody interior. The outer tissue gradually darkens. The size of a crown gall vary from pea size to more than a foot in diameter. The gall tissue gradually rots and there will be growth of secondary microorganisms. It will decay and slow off and new tumor tissue develop in other parts of the plant. In the case of severe infection, severe crown gall, there will be lack of vigor in the plants. The leaves will turn yellow or red and stunted. Shoots often die back. The plants will wilt and die. This image shows plants with crown gall. Coming to disease cycle and epidemiology, Agrobacter tumefaciens is present in soils. These are the causative agent for crown gall and they reach soil from galls, from broken or sloughed off galls from infected plants. The cultivation practices and irrigation aids in the bacterial dissemination. It is also spread by infected and infested planting stocks. The secondary spread of the disease occurs during pruning and also from cultivation equipments. The pathogen enter the plant through fresh wounds which are made either during pruning, cultivating, transplanting, budding or grafting procedures or through the wounds created by chewing insects, nematodes or animal pests. Agrobacterium tumefaciens have a typical plasmid, Ti plasmid, which is tumor-inducing plasmid. It has several genes, virulence genes in it, and these virulence genes get expressed when the bacterium perceive plant phenolic compounds, which are released through the wounds, and there will be formation of a long fluxus filament in the bacterium, which is known as the T pilus. Using this T pilus, the bacteria get attached to the plant cells. And once the bacteria attach to the plant cell, there is a portion of DNA in the Ti plasmid known as tDNA. It will get transferred to the plant cell through a conjugation procedure, and the tDNA, when it reaches the plant cell, will get integrated into the plant chromosome, and then the tDNA genes will get expressed and results in the formation of hormones, plant hormones, auxins, cytokinin, and there will be formation of opines. The opines, there are different types of opines. They act as nitrogen and carbon source for the growth of the bacterium, Agrobacterium tumefaciens. And the auxin and cytokinin results or induces abnormal cell division, cell enlargement, and uncontrolled growth of plant tissues. A small enlargement in the plant tissue is generally observed 10 to 14 days after infection. There will be rapid and irregular division and enlargement of the host plant cells. Along with this rapid division, there will be decrease in the normal differentiation of cells within this tumorous growth. And as a result, there will be incomplete and disorganized water and food conducting tissues. And as a result, the crown gall tissue will be unable to obtain enough water and nutrients, and as a result, the enlargement ceases. Decay of the tissue begins, and the outer dead tissues will start to slow away. There will be breakdown of outer tumor tissues, and during this breakdown, the crown gall bacteria get released back into the soil, where they can be carried in the water to nearby areas and infect new plants. So how we can manage the crown gall? While selecting planting stocks, a visual examination for crown gall tumor is conventionally done as a primary screen so that infected plant stocks are not used for plantations. But this is not that much useful, since Agrobacterium tumefaciens can also reside on soil and also on the soils related or nearby to roots. Another approach is in the site selection. A crop rotation program helps in the management of crown gall. Crop rotation programs having cereal crops followed by green manuring helps to prevent the disease spread. Fields that have grown cereal crops for a long period are generally crown gall free sites. The use of chemical eradicants such as creosote based compounds, copper based solutions, sodium hypochlorite, etc. can be used to prevent bacterial infection. Another important approach for crown gall management is biological control by using the bacterium Agrobacterium radiobacter. Some strains of Agrobacterium tumefaciens are sensitive to Agrobacterium radiobacter. It produces an antibiotic known as agrocin 84, which inhibits the growth of Agrobacterium tumefaciens by inhibiting the DNA replication and cellular growth. So, for preventing crown gall development, plant roots may be dipped into Agrobacterium radiobacter K84 suspension before planting. Another crown gall management approach is genetic engineering, where transgenic crop plants are developed which harbor one or more genes whose products can degrade or inactivate the tDNA strand from Agrobacterium tumefaciens thereby capable of inhibiting the agrobacterium infection in the plant. So these are the various approaches which could be used for inhibiting or preventing crown gall disease spread. Now we will discuss another bacterial disease of plants, that is angular leaf spot of cotton, which is also known as bacterial blight of cotton. The causative agent is Xanthomonas campestris malvacearum. This is a gram-negative, motile, rod-shaped, non-spore-forming bacterium with a single polar flagella. This disease is prevalent in all major cotton-producing areas of the world. The most common and conspicuous symptom of this particular disease is the formation of angular leaf spot. These are generally dark green, water-soaked spots, initially more clearly visible on the underside of leaf lamina. The older spots will become dark brown or black in color and are visible on the upper side of the leaves as well. The angular spots will cover much of the leaf surface. There will be chlorosis followed by necrosis and distortion of leaf lamina. Sometimes the bacterial blight of cotton also causes bacterial ball rot, where there will be roughly spherical water-soaked spots formed on the surface of the cotton ball. These spots will become black as they age and they penetrate the ball cortex to cause internal ball rot. Symptoms of angular leaf spot of cotton. Different parts of the cotton plants are affected. Both seedlings and mature plants will be infected by this bacterium, and the symptoms generally appear first on leaves. There will be small, round, water-soaked spot formation which will turn black and dry up. If the young seedlings are affected, the seedlings may appear green with a black tip. The seedlings die rapidly upon bacterial infection. In less severely affected plants, the spots on the leaves and stems become water-soaked. They will enlarge into angular reddish spots about 1 millimeter in diameter. All these spots coalesce and the leaf gradually yellows and drops. The symptoms on stems. There will be lesions on young stems, become long and black. There will be girdling of the stem, and this results in the loss of fruiting branches, and this symptom is generally termed as black arm. Blackened appearance of affected petioles and branches are there, and thus is this symptom known as black arm. The older stems, spots in the older stems may callus off, and this will result in open cankers. There will be yellowish bacterial exudes from the lesions, generally in moist weather. The cotton balls are also affected by the bacterial infection. There will be formation of water-soaked lesions which will join together, coalesce together to form irregular, large, brown, sunken areas. The balls, when infected very young, the balls will drop prematurely. But if the infection occur in the balls when they are older, then the ball shape it will become distorted. So this is a leaf having the angular spot, and this is a cotton ball having the particular lesion formed. The control measures to prevent the spread of angular leaf spot of cotton is the cultural and sanitary methods such as crop rotation, deep plowing of the land, soil sanitization, and seed treatment with carbon disarm. Infection-free and certified seeds should be used to produce the plants, and any infected plant debris in the field should be properly destroyed. An integrated pest management process can also be adopted for proper control against the angular leaf spot disease of cotton. So, we were discussing about bacterial leaf blight of rice, citrus canker, crown gall, and angular leaf spot of cotton. Thank you so much for listening.